Hello, everyone. A very warm good evening or good morning. We'll be starting with the session in more 10 minutes. Meanwhile, we begin with the session. You can introduce yourself in the chat box and let us know about your background and your research interest. Hello, everyone. I would like to welcome you all to today's session. We'll be starting in more 5 to 10 minutes. We are waiting for other participants to join as well. And meanwhile, we begin with today's session. You could introduce yourself in the chat box and let us know about your background and your research interests. Thank you. A very warm good morning, good afternoon, or good evening in whichever part of the world you are. I would like to welcome you all to today's first session on Introduction to Bioinformatics in R. So before we begin with today's session, I would like to introduce ourselves for those who have joined us for the very first time, and then I would pass on the stage to Dr. Raghavendran to begin with the session. So as the need for management and analysis of data grows, coding is becoming a must-have skill for bioinformaticians. This does not have to be overwhelming career change for a biologist. So our team put together elementary tutorials to understand programming basics using examples of loading, analysis, and visualization of structured data. Our programming offers a complete range of functionality that you can leverage to perform in-depth statistical analysis, visualization, and annotation. Getting started can be hard. Programming is like learning a new language altogether. That is why we offer easy to follow structured and simple coding tutorials designed around bioinformatics challenges. By completing this course, anyone will be able to utilize this popular coding language to create objects. To utilize this, using predefined packages used by bioinformaticians in various research projects. To introduce myself, I am Sonalika Ray and I am the Bioinformatics Project Manager at Pine Biotech. And to introduce our company, we are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools. And our mission is to make bioinformatics more accessible. I would now like to introduce the founders and team members of the company. Dr. Alfred Tauber comes from a medical background with an expertise in oncology, immunology, and biochemistry of information. And one of his major projects is the establishment of Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a result of hard work by some of the people mentioned here on the right. Julia Panov is an expert bioinformatician. Abby Titowski leads the development of the T-Bioinfo platform. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in peer-reviewed journals. He has been developing collaborations with universities as a mentor and project lead for Omics Logic training and research fellowship. Dr. Raghavendran Lakshminarayanan is an accomplished computational biologist with an experience in large-scale analysis of multi-omics data and data science methods in precision oncology and protein DNA structural bioinformatics. So how do Omics Logic resources help advance bioinformatics teaching? Omics Logic is a portal for practical and theoretical learning of bioinformatics composed of courses designed to take you through the theoretical knowledge. Omics Logic takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give a student a clear path up the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic coding languages like R and Python, or beginning a data analysis project altogether. This program comes with an access to the Omics Logic courses, which includes courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, and epigenomics, data science that includes bioinformatics using R and Python. And also the users will get an access to various example projects or case studies, I must say, of different fields which you could learn from and replicate for your research work. Users will also be given the directions to access the T-Bioinfo platform for bioinformatics processing and analysis of different types of data in these different resources that I just mentioned. So without much ado, let's first of all begin by signing up on the Omics Logic Learn portal. I'll just take you through the portal. Once you have signed up, you need to make sure that you have completed your profile. Now, a complete profile will include a profile image, full name with proper capitalization, You must link your social media handles, that is Facebook, LinkedIn, Instagram, or ID, or Twitter, and add a brief bio about yourself that will consist of educational background and research interest. The mentors can view your progress under the Activities tab, and the courses that you are currently completing will appear under the Courses tab. And finally, after the completion of your course, the certificates will appear under the Certificates tab. So, for those who have registered for the program, I would like to take you through some of the things that you have to keep a note of throughout the program. Before I do that, I would paste the link in the chat box so that you all can sign up on the portal. Ujasvi has already shared. Thank you so much. Those who are new and have joined the session for the first time and have not signed up yet on the portal, please take some time to sign up on the portal so that you can get along with us today. We'll be following some resources from the portal itself today and practice some code sessions. It will be helpful to you if you have an account on the portal already. You can take a minute to sign up if you are on your system. Those who have already signed up can put a yes or one in the chat box so that we know we are good to proceed ahead, and others can take this time to sign up. Okay, I can see 2 responses in the chat from the audience. All right, so those who have already signed up, the next step for you is to complete your profile. As I mentioned that we'll be monitoring your progress at the back end. So it's very important for us to know what background you come from and what are your research interests. Knowing them only will help us to align the resources for you. You need to make sure that you have your profile complete, as I just told through my presentation. And after the coursework completion, your certificates will appear here under the Certificates tab. Now, I wanted to go through the program page with all of you to tell you where you would find the resources for the specific program for which we are meeting today. So this is the program page. I'll just put the link in the chat. So this program page is very important for those who have registered for the program. Please bookmark this page. On this page, you'll find the link to join the Zoom session when the schedule is placed for all of these sessions. So you need to just click on the Zoom icon and you'll be able to join the meeting. So you have the schedule here for all these sessions. You have the dates mentioned. Sorry, and you also have the topics that would be covered in each and every session. Now, once the session is complete, you will find the recording of this session on the program page itself. If you're not able to join the session, let's say you have some prior commitments at that time and you're not able to join the session, then you can watch the recording from the program page itself and let us know in Skype group if you have any questions so that we could help you clarify them and proceed ahead. For each and every session, you have some associated online resources which are present here. Now, these resources are lessons and courses on the Omics Logic Learn portal itself. So you need to make sure that for each session that we have put together on the program page, you need to watch the recording. If you miss the session, you can revisit the recording as well to revise the concepts. And then you need to complete the resources that are present here under each and every session. These all resources are hyperlinked. You just need to click on them and you will be redirected to the specific lesson that you are required to go through. In the first session today, we'll be just introducing you to bioinformatics and also introduce you to the language. Some of you might not have a bioinformatics background, so that is why we will also introduce you to the field bioinformatics and the role of big data in bioinformatics before we actually get started. So today will be just an introductory session for all of you to get started, and then we'll dig deep into the concepts. And once you complete the session, you are required to click on the complete session button. Once you click on the complete session button for all of these sessions that we have put together on the program page, doing so will help you get the certificate towards the end of the program, again in your profile section. So that was all about how you can monitor all the resources that we'll be providing you with as a part of this program. This was about the resources. The second thing comes communication. You all are requested to join the Skype group so that you stay in regular touch with our team and the mentor. And whenever you go through these resources and encounter any question or doubt that you might like to clarify, you can ping us and we'll help you with your questions. We'll also have a live Q&A session where you are supposed to come up with all your questions to get them clarified from the mentor. So that was about communication. And now let's begin with the session. I'll now like to pass on the stage to Dr. Raghav to start over with the first session. Over to you, sir.
Thank you, Sonalika, and very warm welcome to everybody. Let's see, let me set up my screen sharing. Can you see my screen and can you hear me clearly?
Yes, your screen is coming up. I can hear you clearly. I can see your screen now. Thank you, sir.
Okay, thank you very much. I'm also following the chat box. So if you have any queries, quickly respond to me in the chat. So most of this is, let's set it up. Okay. Wait a second. Yeah. So welcome one more time to today's session. So we are going to, as Sonalika mentioned already, so we are going to deal with getting started with bioinformatics in R and some of the background necessary information about how are we, what is the kind of data that we are going to deal with when we have to perform bioinformatics analysis in R, and where does this data come from? What are the processing steps that are involved in generating this structured data? So that will be our starting session today. And today also I will show you, when we have time, how to access a couple of lessons, R coded lessons or coding lessons from the Learn portal, and also show you the inbuilt console, R console, that we have introduced or that we have incorporated into the Learn portal itself, so that you can learn and test your knowledge or test pass some of the challenges that we have set for you to understand your learning in the same page itself, or the same web link or same Learn portal page itself. So that is going to be very convenient as we move along. So let's quickly get started. So let's begin with the question, what is bioinformatics, right? So it is, for example, it can be regarded as a convergence of biology and data, where both are equally important. And we will also see in next few slides how the data part and the biology part come into becoming equally important in this field. And let's ask another question. For example, what do we use bioinformatics for? Or a better question is, who uses bioinformatics and who will benefit from these analysis, right? So the answer might be, for example, people who are statisticians or who are computer scientists or engineers. They may be interested in bioinformatics because largely we are going to deal with data. And these are all the people who are associated with data for a large part of their career. But for the last few years, the truth is, like, more and more biologists and more clinicians are actually involved in understanding their data from bioinformatics analysis. It is not luxury now, but it is now expected that biologists and clinicians are able to handle their own data, and they are able to, for example, understand the patterns in the data. And in order to be successful in basic and translational research, we have to use some form of bioinformatics or data analytics for our biological data from biologists and clinicians themselves, right? So this has sparked a transformation which actually transforms the science of biology itself into a science that is rapidly acquiring characteristics of something like data science part. So how is this transformation acquired? For example, we can see that in today's era, biology is about can be defined or can be described as some part of data science, because billions and billions of data points on genes, proteins, and other molecules are actually compiled in a large file, and they are systematically studied. So these characteristics or these characteristics of biology actually makes them very closely resemble the characteristics of data science, right? And these data, right, just like any other data, and analyzing these data by incorporating or by using standard data science analytical tools will lead to more knowledge and will lead to more understanding about the biology of the living organisms, about, for example, understanding the biotechnology part, understanding, for example, crops, livestock, health applications of this data. So, so thus we have actually invented, or actually bioinformatics has become a new field in itself, where it can self-sustain the interpretations and self-sustain the analysis that it is actually creating from the data that are available from biological resources. So this humongous amount of data or volume of data that we will see in a few slides that is being generated from for biological science, for biological understanding of the biology of the sample, has its influence in, for example, development of the associated methodologies and development of some specific resources like approaches that are purely data-driven for understanding biological phenomena or for understanding the biology of a system. And these characteristics, as we spoke about, are actually driving bioinformatics, are actually driving biology to more and more look like data-driven science or data-driven approaches, right? And long forgotten or long before, biology was used as a hypothesis-driven study, where you generally start your investigation based on prior understanding of a system. And then you develop a hypothesis on the relationship that you have understood or that you have knowledge on based on previous studies or based on another portion of experiments that you have already understood. And this hypothesis will fuel data generation and then subsequently data analysis to validate this hypothesis. But today, since the availability of data is large and it's in huge volumes, the data-driven approaches are more and more common, where people start looking for patterns in the data to begin with, right? Even before starting to formulate a theory or a hypothesis, they start to look for patterns in the data. And then once some of the patterns are evident and some of the patterns are not very well defined, so they start making this hypothesis and making this relationship between the patterns that they observe and the phenotypical characteristics, and they can start analyzing this data in a more systematic manner to get to the bottom of this analysis and get to the complete understanding of the biology behind the system. So, we spoke about big data and large volumes of data, right? And where are these data coming from, and what generates this data? And this data is based on, for example, high throughput technologies or next generation sequencing technologies that can generate billions and billions of data points on genes that will become genomics data, and proteins and other molecules that will become subsequently transcriptomics and proteomics and other data, right? And these multi-different omics data are actually compiled into repositories, and they can be systematically studied and understood, and they can be interrogated for establishing trends for many different applications, right? And so where is this data and what's out there, and is such data available for everybody to access? The one short answer to that question is yes, and we have, as a part of biological research, have been extremely familiar with one such repository that is maintained by National Center for Biotechnology Information, and this is one of the most famous repository that has a collection of all the peer-reviewed articles, biological data sets, projects on various topics, and etc., right? So we have definitely used PubMed, where we can see many of published peer-reviewed articles, and we can actually identify data sets from understanding or from scanning through several of these publications from PubMed database itself. And one of the associated resources or databases that we will definitely discuss in the future sessions is the GEO, or Gene Expression Omnibus database, which is a collection of thousands and thousands of transcriptomics data and RNA-seq data and single-cell RNA-seq data and metagenomics data and several other data that are compiled for public access and public analysis. So we will definitely use some of these data sets, and in the next few sessions and in the upcoming sessions, we will also discuss how to download data sets from publicly available resources, and what is this data set, and what are the type of analysis that we can perform from this data set, and how to set up these specific analysis for the data set that you have selected or that we have focused on. So those things will become very clear when we discuss in those specific sessions. So we actually discussed, for example, biology and bioinformatics acquiring properties of data science. So what are these specific properties of data science that are related to bioinformatics data or biological data, right? So to give you a brief overview of, for example, data science is a scientific field that involves developing methods of recording or storing and analyzing data to, for example, effectively extract useful information. So the ultimate goal of data science is to gain knowledgeable insights from any type of data, both structured and unstructured, or sometimes semi-structured. So unstructured data is what you typically would have to deal with when you are trying to acquire data from publicly available resources, right? And this unstructured data can be, for example, understood as loosely organized data of different categories, like your emails, or like your, for example, tweets, and like survey data, which we'll also discuss shortly, right? And this has to be prepared for analysis to make it completely useful, or to extract more and more knowledge from this unstructured data. This has to be prepared, and this has to be structured into some form where we can apply some statistical analysis for understanding inferences from this data. So what do I mean by unstructured data? Let's to illustrate this concept, let's try to, for example, conduct a mock survey. So if I were to ask you what is your educational background, the responses might be extremely varied. For example, some might respond like, I am a graduate student, and some might actually give a very detailed response like, I have a degree in computer science specializing in network and etc., right? And others might actually give acronyms of their degree that they are going through. So these are all varied responses, but I cannot make out a lot of things from these responses. As a human, if I have to go through these resources or these responses, then I get a good idea. But if the number of data points increase a lot, then human element becomes really difficult to incorporate, and we have to rely on analytical methods and algorithmic methods to understand the patterns in this data. So what are the patterns that we are looking for? In order to make it a little bit more structured, we can actually introduce structure into some data collection techniques like this survey, right? By replacing the information with actual options that you can choose from, right? I can give you options based on which I can make some inferences of this structured data generation. But this actually reduces the kind of information that I might be able to obtain, right? So I will not probably cover all of the options that is available there. So hence, introducing structure into the data will actually promote a lot of analysis, which is absolutely necessary, but it can also result in loss of quantity of data or loss of variation in the data. Sometimes it is sometimes which is considered as a necessary evil, right? So we cannot do a lot with unstructured data. So either we take the unstructured data and introduce structure by analyzing the outputs, which again will might lead to loss of data. But generally, this is to get you or give you an understanding that we need structured data to be able to perform statistical analysis and to make some valid and valuable insights from the data, understand the trends of the data, like it's given here. So same can be extended to any kind of data, specifically big data, which is our primary interest here, right? So big data in its at its ultimate or at its basic level has certain characteristics that we have to keep in mind. And a big biological data or bioinformatics data is not an exemption from this. So they are characterized by several characteristics, but some of the important characteristics that we have to keep in mind is the velocity, variability, and volume. So we know that it is the volume is humongous because it we know the quantity of data that we are dealing with is in terms of GBs and GBs. Sometimes it can also reach terabytes, and we know the variability is also a major factor because some of the omics data types like metagenomics data types and the single cell transcriptomics data types are so varied that alternative methods of analysis might not be very fruitful in understanding the patterns in the data. And we know about velocity also because the data keeps on increasing in its quantity every year. So we can see that when we analyzing the amount of data that is deposited to repositories like NCBI and other databases from where we can download these omics data types. So, so that helps us understand, for example, the necessity to structure any data and largely importantly to structure omics data, right? So how can we introduce structure, or how can we convert raw omics data, which is predominantly unstructured, to a structured omics data, which is absolutely necessary for analysis, like that we are going to perform in the following sessions, right? So to introduce structure or to structure omics data, we basically need to take the raw sequencing data, which is in the format of string of letters A, T, G, and C, and to generate a data set or to generate a matrix of continuous or categorical data, which can be analyzed in a statistical environment. So to do that, we have to organize steps of processing algorithm in sequences, which we will refer to as pipelines. And these analysis pipelines are specific to particular type of omics data. So for example, for genomics, we will have to set up a different pipeline of analysis methods. And for transcriptomics and metagenomics, we will have to set up a different combination of algorithms for their pipeline setup. So that's how we can structure any omics data type based on the sequence of analysis algorithms that are available for that to handle that specific omics data type. So so what do we have in the public repositories? For example, if publicly available data is majority, majority of the publicly available data is unstructured, then some portion of that data is also structured and that is also available. So when you simply compare, for example, we have lots and lots of data that is unstructured compared to some portion of the data that is structured and also publicly available. So it is more convenient for us to take the unstructured data and perform the analysis pipeline, an example pipeline that we will also refer to in the upcoming few slides, and then generate our own structured data for the analysis that we are interested in. Why is this convenient? This is majorly convenient because sometimes the analysis methods might get upgraded and the necessity to generate new structured data might be necessary to effectively understand the patterns in the data, right? And sometimes we will get new information based on some updates that has been discovered or that has been unveiled. A major source or major example for this will be the difference between analysis that is performed on the HG19, a previous version of reference human genome, compared to the HG38, a newer version of human reference genome. So the newer version will obviously include mapping of sequences to different portions of the genome, and then definitely will give us information on this new location or new genes and new genomic positions that are not covered in the previous version of analysis that uses the previous version. So it's much better if we are going to consider, or we will consider, unstructured data and then devise our own analysis pipeline with the latest available algorithms and latest available reference genomes to arrive at most updated pattern that we can use for downstream analysis that we will definitely discuss in the future sessions. So to achieve this specific result, for example, to or achieve structuring of the data, we will usually take FASTQ files or raw data. Let's consider genomics data, right? Let's consider genomics data. So to structure genomics data, we will take raw FASTQ files and then run a pipeline, for example, called variant calling pipeline. This pipeline will give you an output of all the list of variants that one can identify in the sample that is being sequenced compared to the reference genome that we just discussed in a short while. So the pipeline generally might include several of the steps that are categorized as pre-processing steps, mapping algorithms, and then followed by analysis of these mapped regions like somatic variant calling steps to produce the list or to produce a table that we can perform downstream analysis with. So for example, when we use the FASTQ files that is generated from shotgun sequencing or high throughput NGS sequencing like Illumina, right? We will generate lots and lots of short reads of sequences that are sequenced from randomly selected fragments of DNA or RNA. So generally, these fragments will have to be aligned to the reference genome, and once aligned to the reference genome, and these short reads can be analyzed for mismatches and non-matches or deletions to the reference genome. These mismatches are the ones that we are going to characterize for the data in the event of genomics data analysis for the sample that we have sequenced these reads from, right? And these mismatches or non-matches, for example, in oncology, let's say in oncological context, can be analyzed for their mutation status, which are often referred to as somatic mutations, or mutations that are observed in the tumor tissue compared to the normal tissue that is very closely situated in the biological sample. And these list of mutations will be very important to understand how they might affect some of the important genes that are involved in, let's say, tumor growth or tumor disease progression, etc., etc. So to analyze such a data, we can prepare a table of somatic scores for each and every genomic position, and that table can be fed into, for example, more detailed or more advanced downstream analysis that can also involve statistical tests or can involve machine learning analysis to identify genes that are associated with statistically relevant somatic scores compared to the normal. So this is one way where we can go from raw sequence data to a table or a list of structured data, and then subject that table or list of structured data into statistical analysis and other advanced statistical analysis to extract meaningful insights from this data. Right? So another example that is mostly referred to in many of the publications is the transcriptomics data, and this data represents the mRNA expression, which is a good way to analyze what are the processes that are going on inside the cell by observing the active transcription of the protein coding genes. Right? We all already know by this stage that the DNA is transcribed into RNA, and the RNA is what is translated into proteins. So by measuring these messenger RNA, we can understand what are the proteins that are essentially available in what abundance in the cell's particular state. Right? But for example, this method or this transcriptomics data analysis will also give additional information than just giving you quantification of genes and their expression levels. For example, the protein coding genes in the genomic DNA that we already know may contain large stretches of non-coding sequences that are called introns, right? And we also know that some portions of these introns are actually spliced, or not some, actually, most of these introns are spliced out of the RNA transcript by spliceosome, and these splicing can actually change the constituents or change the parts of the gene that may result in one or other isoforms of that same gene. So these isoforms may actually be lead to or may be associated with, for example, the diseased state or altered state of the cell, and their expression levels can be analyzed and associated and generally linked with the altered state of the cell that we are interested in characterizing. Right? So a typical analysis for, let's say, transcriptomics data or RNA-seq data, no, both are same. Transcriptomics is, they might include other forms of quantifying messenger RNA like microarray, right? And targeted sequencing. But RNA-seq is just one next generation sequencing technology involving a method to quantify all the messenger RNAs that are present in the cell. So transcriptomics is the umbrella, RNA-seq is a major technique which generates large portions of this transcriptomics data. So before this RNA-seq was involved, transcriptomics analysis was performed using microarray sequencing or microarray technology, in fact, right? Not sequencing, microarray targeted probing technologies. So that is the difference. So, but nowadays it is actually used synonymously, because majority of the transcriptomics data analysis is done using RNA-seq platforms. So a typical analysis pipeline for the data that comes from this next generation sequencing technology and our RNA-seq data, for example, will allow us to again map the, okay, what happened? To allow us to, for example, pre-process the input files, right, the FASTQ files, and then clean for any presence of adapter sequences and etc. And also clean some of the PCR duplicates that are found, and then we have to map these pre-processed reads onto the respective genes in the human genome, and then quantify these mapped reads or quantify these genes based on the number of reads that are mapped onto that specific genomes. This can often, or this will often be followed by downstream analysis that involves estimation of differential gene expression, which identifies genes that are expressed specific to one type of sample compared to the other. So as a result of this quantification of this RNA-seq data, you will be able to obtain a table of continuous numeric data that is annotated by gene ID and organized consistently across different samples in a given experiment. So you will have, for example, along the columns, the expression levels that are quantified for different samples, and along the rows, you will get the expression levels that are quantified for different genes as given by their ensemble ID in this case. Yeah. So at their intersection, you will get expression levels for the type of normalization that you have chosen. In this case, it is FPKM or TPM values, and you can also choose normalized count or raw counts or and RPKM as other options of normalization that you can obtain from this pipeline of analysis. So this structured data can again be directly used to, for example, perform a simple statistical association and analysis test, where we can use t-test to find out, or t-test to understand gene expression levels of those genes which are different in, let's say, altered cell state compared to the control cell state, right? This can be elevated, or this can be further applied using specific algorithms such as DSEC2, which performs the same differential gene expression analysis using convoluted or using more statistically defined methods to identify differentially expressed genes that are expressed specific to one condition compared to the other. This can again, for example, be used to perform advanced statistical analysis like machine learning analysis, which can identify genes behind, for example, behind categorizing tumors into their subcategories, or genes behind tumor progression and their treatment response, right? Depending upon the data and more sample characteristics and clinical characteristics of the samples that we can accumulate, we can keep varying the kind of downstream analysis for the question that we are interested in from such a wealthy of data. And another type of data that we will definitely review in this course comes from same sequencing technique, the next generation sequencing technique, but it does offer a completely different view of the biological sample. We will deal in detail in some of the resources, like the RNA sequences are basically the reads have to be cleaned for the presence of duplicate sequences and biases. So the cleaned and trimmed reads to maintain high quality sequences, right? Reads are then mapped onto their respective positions in the genome. So what are the respective positions? Since the data that we are involved with is transcriptomics data, so we are going to get reads only from the messenger RNA or only from the exons of these genomes. So you take the reference genome and then map these reads onto those specific portions, right? Specific genes. And this can form the basis of simply counting the number of reads that is present or that is aligned to that specific gene. So this is very oversimplification of how these reads are converted into counts, but the algorithms such as, for example, HT-seq or RSEM will perform expectation maximization processes to correct for the length of the gene, which will obviously have a positive or have a direct correlation with the amount of reads that can be mapped. And for example, biasness that are introduced generally to certain portions of the genome that is prone to getting a lot of fragments generated at the library preparation stage. So at its simple level, converting RNA sequences into counts is just counting the number of shattered reads from that specific portions of the sample that can be mapped onto any particular gene in the reference gene. And at this complex level, this will actually involve cleaning up, mapping accurately, and also to perform expectation maximization algorithm to account for some of the biases and other variations that we observe. So let's come back to this metagenomic sequencing data. So as I mentioned, this offers a completely different view from the same sequencing technique that we have been discussing so far, the next generation sequencing technique or high throughput sequencing technique, right? So in this case, we are looking at sample that contains millions and millions of microbes or microorganisms that are represented by genomes that are very different from each other, right? So to structure this data and to organize this hugely varied genomic information into categories, we can take, for example, a functionally related gene that is common to all of the organisms, right? Like the ribosomal gene, which can be compared to a databases of known sequences of ribosomal genes and their taxonomy identified. So that is what is roughly being done in the metagenomics sequencing data. So this ribosomal gene that we are going to consider is the 16S ribosomal RNA gene, and the sequencing and the analysis that we are going to perform from this from sequencing this 16S rRNA gene is commonly referred to as amplicon data analysis that can be used, that can be done in, for example, in the Turbot Bioinfo platform using two options, DADA2 and also using KIND2 analysis. So as a result of this, you can, for example, obtain proportions of taxonomic groups of samples that can be analyzed to understand sample diversity, or that can be analyzed to understand the proportionate abundance of specific microorganism that can, and this proportionate abundance of specific microorganism, like let's say, in this case, you can generally see in this output the pink portion of these bar plots, which refers to Vericomicrobiaceae, and that are abundant in only these samples and that are not at all found in these sample, can be linked to the phenotypical variation and can be linked to the to understand the biology behind the sample that we are trying to characterize. So this is the end result, but what is the structured data that you will definitely, or that you will generally deal with and that you will generally obtain from metagenomics data analysis is, it is also a table of quantified data, and the quantity that is being quantified is the operational taxonomic units, which are actually tabulated for their abundance levels across different samples, right? We have again the samples organized as columns and operational taxonomic units, which can refer to specific or which can refer to more closely related microbes that can be identified with their taxonomic ranks, right? So OTU1 and OTU2 and 3 are respective, can be understood as respective microbes that can be whose identity can be understood by comparing, or whose identity can be estimated by comparing the known database of 16S rRNA and their taxonomic association. So this is what is being done in the metagenomics data analysis, where you get two kinds of information. At the first level, you will get this abundance table or abundance information, which tabulates or which quantifies the abundance of these taxonomic units across different samples. And in the second layer of information, we will get the taxonomic identification of these respective taxonomic units, and hence we can make inferences like what we actually discussed in a couple of slides ago, where we can specifically identify the kind of kingdom, phyla, or class level organization of samples that are proportionately abundant in one particular group of samples compared to the other. Right? This is what we can actually do. And to perform such analysis in majority of the programming environment, by now you would have understood that the requirement is simple. We need a table. Right? That table can come from genomic data analysis, it can come from transcriptomics data analysis, it can come from metagenomics data analysis, it does not matter. So we need a table, and that table can be read in as, for example, matrix or array or data frame into an environment like R, right? And there are differences in how these different objects, as I just mentioned, matrix, array, and data frame, are actually going to deal or actually handling data of different class or different types. For example, matrix will load or will actually consider data that are only numerical data types, right? Array can consider again data of consistent data types, right? They're not only numeric, but they can also contain data that are particularly character or one data type or one form of data type. There is no mix matching of data types in matrix or array. Vector is just one dimension of matrix, right? Only one. But very useful object that we will use multiple times in the R data analysis is the data frame. So as depicted in this picture, a data frame can is an object which can be used to store the tabular information or table of data, like we have generated from by structuring different omics data. But data frame gives us a flexibility of storing not only numeric data, but it can also store categorical data, it can also store logical data, it can also store character data, and etc. So this is very convenient because our data so far that we have structured will involve, for example, at least one column of character data that refers to the identity of either operational taxonomic unit or gene IDs, or for example, genomic positions, right? Or genomic annotations. In this case, position is usually numeric. So annotation that can be loaded in along with the numerical data that characterizes their levels across different samples. And some other useful objects that are present in R is usually list, which is just a kitchen sink of all the other types of data that we can put in. And they are very useful because more often than not in our analysis, we will generate different types of data, and we can organize all of them into a list that can be used to, for example, load a variety of different types of data with variety of different dimensions, which is not actually possible in the other types of objects, right? All the other data frame, matrix, and array should have consistency in their dimensions. All the rows should have equal number of columns, and all the columns should have exactly equal number of row entry in the rows. So that is absolutely necessary. So we will also deal with this a little bit more detailed in the upcoming sessions, where we will load tables into R's environment and see how it is actually referred. So we will also quickly do that today. So what does this allow us to do? Right, that's the main important part. Why should we do this? Right, by doing this or by loading data into environment like R will actually help us to perform some statistical analysis to basically learn about the patterns in the data. We can, for example, perform or plot histograms to understand data frequency. We can perform scatter plots to understand data correlation between samples. And there are many more different ways to explore and characterize large amounts of data that we have usually in the data analysis. So understanding this data, then we will actually use several of the representation that we actually use, like scatter plot or histogram, to, for example, visualize the data and to spot technical noise level or to spot a specific threshold with which we have to perform some very important statistical analysis that is required for our downstream analysis methods. And this might involve something like normalization using quanti-normalization method. This will inform data transformation steps like converting the scale into logarithmic scale, and several other transformation and explorations will be discussed in the next session immediately. Right? Yeah. So we will also discuss how this table of data can be converted into, for example, representation like this, and which is more, which gives us more biologically meaningful inferences. Like we can immediately understand the separation between sample based on, for example, the distances between the sample based on the data variance as observed in the PCA plot on the right, or the grouping of sample according to expressions of selected few genes in the form of a heat map as represented in the plot on the left. So these are all only starting points, and it will give us hints, and it will give us pointers to a right direction into what are the samples that we can compare and how we can set up this comparison, right? Those will also be coming to play. So these exploratory data analysis and visualization in multiple different options will give us a chance to see how different methods like data mining methods and how different machine learning methods can be used to group data objects by their expression levels, or by their taxonomic abundance, or by their mutational frequencies, to generally understand the similarities and to differences between the samples that can be related to their phenotypical characteristics or biological conditions of the data, right? And we can also, for example, use this data for applying supervised machine learning methods, which can learn or which can create a model from the data that we have supplied, and then make more predictive prediction of the data that are observed in the observed in the real world data examples. For example, to expand on this, we can take a group of, for example, we can take a transcriptomics data analysis of a specific cancer type, and then perform supervised machine learning analysis to understand the stages of different tumor types, and then use a completely different data set from the real world data to apply the prediction, or to validate the prediction of the model that we just prepared or we just created using the input training data set that we supplied to the supervised machine learning model. So these are all different types of downstream analysis, and the choice of which analysis to be performed to infer what impactful inferences that we have to take will actually depend on what are the exploratory data analysis points to from our data. So that is why you will often see us discussing the importance of performing very useful exploratory data analysis and visualization upfront when we are exploring the patterns in the data before setting up specific analysis using statistical and advanced statistical methods that we will discuss. So if you have few more minutes, I will reshare my screen, and I want to show you some associated resources in this case to tell you how we can access this data from the Learn portal. So that is going to be very useful for us. Let's say, let me set up the sharing. This won't take more than, let's say, 5 minutes. Yes. My Zoom is still working on sharing my screen. I hope you can see my screen. Can somebody reply me that whether you can see the R coding course 1?
Yes, Dr. Raghavendran, we can.
Thank you very much. So this is the associated resources, and Sonalika has actually gone through these resources, so I won't go through again. But several of the resources are associated resources can be accessed from here, and nothing stops you from accessing what is going to be discussed in the next session, right? So I'm taking some of the examples from the next session, like this data processing and visualization and reading DNA sequences in R, right? Those can be very useful if you want to get generally good introduction to R before we even start, right? So one of the very useful course that we will refer time and again over the course of this program is the R coding course 1, and which is very, very valuable for beginners who wants to get started with R. So I want all of you, if you have access to this platform, to go to this lesson, and I will paste this in the chat. Log in and go to this specific lesson. If you are logged in, if you click on this link, that will open up the resources for you, and you can follow along with me. So what am I going to do? I'm going to simply access loading data in our lesson. Then opening up this, will start with an introduction to how to set up R and what is the use of R, right? You can go through this video resources to set up or install R for your computer. But even before that, if you go down or if you scroll down, working through the codes that are given in this, not only in this specific lesson, but in almost all of the R coding course, doesn't require you to install R in your computer. So it is actually platform free or background free. You can access these lessons from anywhere, right? So how do we do that? So we go to learn the code section, where the basic idea of this specific lesson is to take the table that we have been discussing so far and load it into R's environment, and then reproduce and print out the table to see how they are being interpreted, right? So this is how we are going to break down the code. All the codes are going to be shaded in black, and they are copy pastable, and we will define, or we will actually go through each and every portion of these syntaxes at its fundamental level. For example, just going through this one line will actually introduce you to, for example, the function that we need to load the table, and what are the requirements of this function. This function at least needs to be told where the data is, right? Where the input file is. That input file can be located in your computer. It can be located in the internet that is publicly available, and followed by some of the characteristics of your input table. Whether or not in your table do we have title for each and every column, that is the header. So whether or not that is present, and how are these columns separated, and what is the character that separates this column. The character can be tab, the character can be comma, that character can be semicolon in some specific cases. So these are all characteristics of the input file that we need to know before even loading this to R. If we don't provide this to this function, the function will assume default values, and if that default value does not agree with our sample or with our table characteristics, the table will not be interpreted as it should be interpreted. So that is what is given here. You can, for example, extend this to not only from text file in tab delimited format, and we have given you how to import Excel files into R, right? So reading Excel files into R requires a module or requires a library to be installed in R's environment. So if you are going to use your own version of R, so you have to install this library into R before even using this function read.xlsx. Right? That's there. And reading a comma separated version of the file is just using a different function instead of a read.table function that we have been using, right? So after going through this, and after, for example, going through some other portions of how to check whether the data is loaded or not, usually we take this function and their output and redirect that to an object that we can access and that we can check or verify. So that object is usually or always a data frame when we use these functions, right? So that is why they are abbreviated as DF. And R can use this operator, which is simply redirection operator that takes the output of this function and puts it into this object, right? And we can again now verify the contents of this object by simply mentioning their name and then executing in the R console. So that is what is given here, how different ways you can verify the contents of the data frame and make sure that all the necessary rows and columns are actually ready. So to try or to test what you have learned so far, we go to the next section, try it yourself. This is an R console, and that is loaded in the backend of the Learn portal. So you will get to run R commands and R codes in here. So this is not very straightforward. It is straightforward in that case, just to press run, it will run. But I want you to read these instructions where we have prepared this console to help you test yourself what you have learned from the previous section. So how have we done that? So read this. Here we say that we have deleted some important portions of the code and that you need to complete by understanding how it is being done in the previous section. So if we do that, let's say, if I don't complete it and run it just like that, it's going to throw me a lot of different errors, right? It's going to throw first many errors that are not easily readable, right? That's how programming languages are. So it says we need to actually, for most of these codes, we need to take the files link that is given here, right? This is the link, and we right click on this link and copy the link address, and to replace this portion, which is symbolic, and then I right click and paste. Okay, so I mentioned, or I'm giving the function where to find the data set from, and it should run now. So I want to clear this console. Let's clear this and then run it. Again, it's going to throw you an error because now I notice that the function which reads the table or which reads a file is not complete. So I complete the function, then clear it again and then run it. Now I get a success in running the code and the answer is correct. So I also get the output right here. And you can do the same for using libraries also, and that is loaded in the backend. And this is actually giving, okay. So now it looks complete. The function looks complete, and the path where the file is located is not complete. So again, change this, copy link address, and then change this. And right click and paste it. And now it should be good to go. I can run. Let's see. And please observe what I get. Yeah, I get success in running the code, but the answer is incorrect. So there is no syntax error in my code, but the intended answer is not met. So what is that? The intended answer is here, right? We have to check the, we have to print the data, import and view the data. So the printing portion of the data is not there. So just like the previous case, I just print what is given in the data frame. So there is nothing to be clear from this console. So I just run it again. So now I get success in running the code and the answer is correct. And we have given some more examples and some more extra biological data sets for you to try. So that is for you to try. So before closing, we'll go back to the lesson and want all of you to also explore the second lesson, which is going to be the topic for the next session, where this console not only can give you, for example, not only can give you outputs in the given output area, but it can produce graphs like this. And how to produce graphs like this is what explained in this code. That is what is the topic of our next session. I quickly want to show you here. I see that the input file is already filled up. So I complete the syntax. And I see that there is another portion of the syntax that is missing. And true. And then say this, come back and see this portion of the missing, and see this, and then then run this. Correct. And this is not going to produce any plots because that's not the intended use, but this will produce the plot. And let's see. Okay, the codes for producing box plot is incomplete. I can type box plot correctly, and then heat map. Then type histogram, and the data is missing from the histogram df, and then run this. We will discuss in detail in the next session, but I just want to show you that this can produce graphics just as what is discussed, what is described in the lesson or associated resources or learning the code section. This can produce exactly the same graphics that you can generate and then explore the contents of these graphics and to understand the patterns in your data. So with that, I will conclude my part of today's session. And if you have any questions, I'll be happy to take it from the from the chat box, or if I will pass it on to Sparsh. I think, yeah. Sparsh is going to be the next person who is going to talk about.
Yeah. So sorry. Actually, I was unable to unmute myself.
Unmute, yeah. Sorry.
Yeah.
Okay.
So thank you, Dr. Raghavendra. Dr. Raghav, we have two questions. I suppose you haven't answered. I'll just read out those questions. So the first question is there a difference between RNA-seq and transcriptomics?
I actually answered this during the
Okay, so you
session
have
itself.
answered this question.
Yeah, I have
Yeah,
answered.
okay.
Yeah. And
Perfect,
the
perfect,
next
sir.
question also, could you please say that again? RNA sequences according to counts. I think I answered when
Yeah,
yeah.
perfect, sir. Thank you so much.
Any other
I
questions?
just wanted to confirm.
Do we have any other questions? So yeah, if no, then yeah, Sparsh, you can take it over. And if yes, you can type it in the chat box while Sparsh is explaining you.
Thank you so much, sir. So, hello everyone. My name is Sparsh and I'm the marketing manager here at Pine Biotech. So I won't take much time of yours because it's already we are running late. So I'll just quickly tell you about the registration and the resources which one would have access to if they would enroll for this program. So by enrollment, we mean that there is a particular fee structure that you need to pay so as to get enrolled in this program and attend the further live sessions of this particular program. So this is the program page, and let me just share the link here in the chat box. So this is the program page. After the session, you all can go through this program page and take a look about the sessions, program overview, etc. So the name of the program is Introduction to Bioinformatics in R, Getting Started with R. So we'll be covering here about multi-omics, how one can process the data, statistical analysis, data visualization, etc. So today was the very first session, and it was a kind of an introductory session. So we covered all the resources which one could have access to, how the associated online resources would work. Everything was explained you very extensively by Dr. Raghavendran as well as by Sonalika, how to sign up on the portal, update your profile, how to navigate on the Omics Logic Learn portal, etc. So this is again the program overview, as you can see. And when you'll scroll down, you can see a full-fledged curriculum for all the sessions which we'll be conducting, and maximum of the sessions will be live sessions because it includes coding. So those sessions would be live. So the first session we covered includes introduction to big data in bioinformatics, introduction to bioinformatics languages are using the code playground, loading data in R, etc. And Dr. Raghavendran show you the associated online resources on the Learn portal already. So I won't be taking it again. I'll simply tell you about the pricing part. So first of all, there is a note. There is a drop down menu here at the right top corner, which through which you can change your currency according to your location. So if someone is living in a US region, so they can change it into USD. If someone is living in Brazil, so they can change it into Brazilian currency. If in India, then INR. If in Nigeria, then NG and Naira. So for now, I'll be choosing INR, and I'll be explaining the resources and the subscription accordingly. So there, as you can see on my screen, there are three different tenures. One is of 45 days, then is of 60, and then is of 90s. The common resources which one would have access to when they will enroll in this particular program, it would be online tutorials, project examples, coding sessions, session recordings, online mentor support, online technical support, then example cloud pipelines on Bioinfo server, which is our analysis platform. Then after the completion of the program, you will have a certificate also. You'll earn a certificate which you can surely use so as to upgrade your CV and to show the recruiters that you are, yes, you are skilled in our language integrated with bioinformatics and how you can analyze biomedical data through it. And also you can showcase in different social media platforms like LinkedIn, etc., for better and for boosting up your networking also. So these are the common resources. Now, what is the difference between the three tenures is, in 45 days tenure, you will go through with the full-fledged training, and like you can clarify your doubts in those live sessions which will be conducting or via the online mentor or technical support through the Skype group which Sonalika told in the beginning of the session. Now, the difference between the 60 and the 90s is, in 60 days with 45 days of training, you have two different opportunities. One is the one-on-one mentor session. So if while going through your training part, you get stuck in like you have a query and you get stuck somewhere, you can ask the project manager or the community manager to fix a call of yours with the mentor of 15 to 20 minutes, kind of an exploratory call. You can get on a call with him one on one, clarify your doubts, and then move on further with the training part. The second opportunity is to draft a research proposal and present it to the mentor for further evaluation and expert feedback. And the outcome for 60 days tenure is a poster presentation. So you can create a poster of your research, and you can present it in different international conference and research symposium. And also we conduct biannual research symposium, so you will be surely getting a chance to showcase your research idea in those symposium. Now, in 90 days tenure, you have another two opportunities. Obviously, one is one-on-one mentor sessions, and second is to complete a full-fledged independent research project, to develop your own independent research project. And the further outcome of this 90 days tenure is a research publication in a peer-reviewed journal. So this was the difference between the three tenures. Now, talking about the pricing part, the 45 days, the pricing of the 45 days is 7,500, then is 10,000 for 60 days, and then 90 days it's 15,000. And right now we are providing with 20% scholarship. So the program has just started. We'll be ending of the scholarship part today itself. So please go and avail the opportunity so as to be a part of the program. So this was about the pricing in INR. Now I'll change the pricing into USD to showcase you what exactly the pricing in USD dollars looks like. So the first for the 45 days it's $150. Sorry, it's $150. Then for 60 days it's $300, and for 90 days it's $450. Again, we are providing with 20% scholarship on all the tenures. And to avail this, Shubham or Ojasvi Shubham have shared the steps to register in the chat box. You need to simply visit the link here. I have shared, and also Shubham has reshared it. You need to choose the tenure according to your wish and your requirements, whether it is 45, 60, or 90. And after choosing the tenure, you need to click on this buy now option. So when you'll click on this buy now option, you'll be redirected to the payment checkout page where you need to simply write the coupon code, type the coupon code, that is take20. Click on this apply button. Your coupon code is successfully applied. You need to fill this user details and click on this checkbox and select the payment mode which you want to apply for, either Stripe or PayPal. Then fill out your credit card details and proceed with the payment checkout. So this was the steps to register for the program. And now, if anyone is having any doubt regarding the scholarship or the registration or the payment procedure, please feel free to ping in the chat box and we'll be here to assist and guide you. Thank you. So I guess I have converted the price bundle to US dollar already, and the price for US dollar is for 45 days it's 150, for 60 it's $300, and then for 90 it's 450. And I suppose it's clear now. Thank you, Jasvi, for the answer. Shubham has shared the steps to procedure enroll. Do you come with your own project and data, or is provided Omics logic? So it's both ways. You can also come up with your idea, your research question, and our mentor will guide you accordingly. We'll be starting with the training part, and then we'll be guiding you how you can review our literature, find a good data set on which you can work on, prepare a proper methodology, etc. So it is both ways. You have a research interest and you don't know how to develop a research question, we'll be guiding you that also. If you have a research question, we'll be guiding you that also, how you can further develop a full-fledged research project out of that research question. So it's both ways. Okay, so Dr. Raghav Vedrat has also answered this question. Thank you, Dr. Raghav. How can we apply for the scholarship? So we have provided you with the coupon code already, take 20. You need to simply like click on this buy now option, and you need to follow the steps to register and then apply this 20% scholarship. If you are asking for more scholarship, then please mail us at marketing@omicslogic.com. So our team will surely revert back to you and will provide you with a suitable solution which would definitely help you out, and you can surely enroll in this particular program. Also, we have an official number and it's active on WhatsApp. So it's 9876134120. So you, yeah, thank you, Shubham, for sharing the details. So this is the mail address. If someone is seeking more scholarship than 20%, they can mail us out. And this is the contact number. It's an official number. You can contact us via WhatsApp. We can have a direct WhatsApp telephonic conversation also, and we can discuss it in more extensive or comprehensive manner. Thank you. Any other question? Anyone? Sorry. So I guess there are no more questions, and we are good to end this meeting. So yeah, so thank you everyone, and we are looking forward like for the next session, basically session 2. Okay, so okay, Aya is asking one question. For option 90 days, you mentioned it includes publication. Who will be working on the manuscript beside the first author? The person who worked on the project. So yes, Aya, and I guess Dr. Raghavendran would be the correct person to answer this question. Dr. Raghavendran, could you please unmute yourself and answer this question?
Yeah. Okay, so who will be working on the manuscript besides the first author? Aya, it's basically you and me, and if you need any technical help from Sonalika, then she will also chime in, right? That's how the publications are actually made. If the work involves only you and you are able to perform this analysis without much of the other helps or other counterparts, then it's going to be you as a first author, and yeah, and me, and maybe, yeah, that's all, nothing else. So if yeah, and Ilya might be also the last when we are going to discuss with him for necessary inputs for making this publication into a good shape. Or if you want to just learn the methods and apply the methods yourself and then use publication. So we can discuss about the authorship at a much later level. The idea or the intent is to make or help you with reasonable projects that you can showcase, that you can publish, and that you can use to learn the techniques and necessary details. So that's what it is. So it depends on how much of work we did. If it involves me working on the project, then of course you have to share the authorship. And if it involves me just guiding, then it depends upon how much, just simple mentorship, then you can just mention that thanks for discussion with Raghavendra, etc.
I hope your question is answered, Aya. Thank you. Anyone is having any question, please feel free to ping in the chat box, or you can unmute yourself and ask. That's completely fine. Okay, so there are no more questions and we are good to end this meeting. So thank you everyone for joining the first session of Introduction to Bioinformatics in R. Hope you gained something new today. And we'll be looking forward so we can have maximum participation for this particular program, and we'll be looking forward so that you can join in the next upcoming session, that is session 2. Okay, there is one more question. There are courses that require a commitment, so I could not pass them. Are there opportunities to get a scholarship? Ella, yes. So there are some courses which have been marked as starred, and you cannot pass out like those courses. Definitely, yes, we are providing with scholarship, and that is what we discussed, that you can mail us at marketing@omicslogic.com, and we'll be discussing about the scholarship. Also, the second thing is, uh, for those courses, to have access to those courses, there is our starter subscription which you can have access to. Let me share my screen and I'll show you. Just a moment. So if you will click on this pricing after you have signed up, and if you'll click on this pricing tab, just a moment, it's taking time to load. You can see here it's taking time to load. Yeah, perfect. So this is what I was talking about. This is the starter subscription, and in the starter subscription, you would have access to all those courses which you are saying that you are not eligible to go through with those lessons and courses because they have been marked as start. So you can click on this buy now and proceed with the payment checkout and have access to all those start lessons. Thank you. Do you have any further question, anyone? Okay, so I don't think so now we have any further question, and it's a final goodbye and thank you to everyone. Thank you everyone for joining. Take care. Have a nice day ahead. Bye-bye.